Today we will see the structure of bacterial cell. A typical bacterial cell remains surrounded by a outer layer or a cell envelope which consists of two components, one that is the rigid cell wall and second one that is the cytoplasm or plasma membrane. Cell envelope encloses the protoplast. It is made up of cytoplasm. Cytoplasm inclusions such as ribosomes, mesosomes, flat globules, vacuoles, nuclear material. Along with these essential components, the cell envelope in some bacteria may be enclosed in a slimy layer or capsule. Some bacteria carry flagella, fine hair-like fibrin, or pili are also present in some bacteria. We'll see one by one. That is the capsule, microcapsule or slim layer. In some bacteria secretes a viscous material around the cell surface, particularly in a pneumococcus and many other forms. This viscous material organized into a sharply defined structure. It is called as capsule. It is so thin called microcapsule when a viscous material is present in the form of loose under marked secretion as a leuconostoc. It is called slim layer. All bacteria do not possess all these three layers. It is generally polysaccharide or polypeptide in nature containing a single amino acid. The microcapsules are composed of proteins, polysaccharides, and lipids. After completion of capsule, we will switch over to the cell wall. It is extremely thin, 10 to 25 nanometers thickness. It provides a rigidity and definite shape to cell. It can be viewed under the electron microscope. Chemically, it is composed of mucopeptide, that is the murid, and scaffolding or platform formed by N-acetyl glucosamine and N-acetyl malic acid molecules arranged in alternating chains. According to Peberdy, in 1980, only the compound present in the cell wall of both gram-positive and gram-negative bacteria is peptidoglycan. The cell wall of gram-positive bacteria contains a 95% peptidoglycan and 10% teichoic acid. On the contrary, the cell wall of gram-negative bacteria is more complex in composition of several layers just adjacent to cytoplasmic membrane. Inner layer, it is made up of peptidoglycan. Between peptidoglycan region and outer membrane is present a periplasmic region. Several enzymes are present in a periplasmic region. The outer membrane is connected to peptidoglycan layer by many bridges. After completion of cell wall, there is a presence of nuclear material. It is present in each bacterial cell. No nuclear membrane or nucleolus is present. That Bowen in 1976 and Clippy in 1979 suggest that a low electron density region in the cell are actually the densely packaged DNA regions are called nuclear bodies or nucleoids. Such a nucleoids, it is made up of DNA. Such a nucleoid is associated with a mesosome in a gram-positive bacteria. Particularly, the bacterial chromosome is equivalent to its DNA. The genetic material of cell. Recent findings indicates that the considerable amount of RNA and proteins are also associated with folded DNA in a bacterial nucleotides. The DNA of the E. coli is a circular and highly folded. After completion of nuclear material, there is a presence of Another part, that is the plasmids and episomes. Some bacteria also possess some extra nuclear genetic element. It is made up of DNA. These cytoplasmic carriers of genetic information is called as plasmids or episomes. But when an extra chromosomal genetic element consists of DNA situated in a cytoplasm in the free state and reproduce autonomously, called as a plasmids. But those genetic elements which exist in a one or two alternate states, either autonomously in a cytoplasm or in the integrated state attached to the chromosome of bacteria, it is called as episomes. After completion of plasmids or episomes, the another part which is present in a bacterial cell, that is the flagella. These are long, fine, hair-like locomotory appendages found commonly in a rod-shaped and spiral bacteria. The presence or absence of flagella and arrangement and number are the characteristic for different genera. There are two broad types of flagellar arrangements. One, that is the polar, and second one, there is the peritrichous. When the flagella absent, it is called as atrichous. While we will see the polar, when flagella are situated at the one or both ends of the cell, are called as polar. When a flagella are situated at one or both ends of the cells, are called as polar. uh, on the basis of that flagellas, there are four types, that is the monotrichous, lophotrichous, amphitrichous, and peritrichous. We will see the monotrichous, that is the single flagellum at the one end, particularly in vibrio coma. Lopotrichous, that is the cluster of flagellas at the one end, particularly in a spirillum. Amphitrichous, that is the cluster of flagella at each end, and a peritrichous, that is the flagellas arranged all over the cell, particularly in typhoid bacilli. After completion of flagella, the another part or structure which is present in a bacteria, that is the fibrin, or it is also called as pili. They are very fine hair-like surface appendages found in a gram and negative bacilli. These are shorter and thin than flagella. That's why it is called as pili. They are only about 0.5 micron in length and less than 10 nanometer in thickness. They are originated from cell membrane. The function is adhere the cells firmly to the particles of the various kinds. Some pili are longer, other pili, these are called as a sex pili. Thank you.